This lecture is on Chapter 3, Cell Structure and Genetic Control. We'll spend some time on the plasma membrane, and then we will go quickly through the cytoplasm and organelles and get to the gene expression. In your course objectives, they focus mostly on the plasma membrane and the gene expression. Okay, so let's start with what the plasma membrane is and what the cytoplasm is, as I mentioned, and what gene expression comes from. So there are three principal parts of the cells: the plasma membrane, the cytoplasm, and the nucleus. And the plasma membrane is selectively permeable. It gives form and separates the cell from the external environment. The cytoplasm and organelles are the fluid part of the cell and the little organs that do the functions. This is what won me over to be a biology major, was the fact that cells themselves have little organs. That's what organelle means, little organ. The nucleus contains the DNA, one or more nucleoli, and it directs cell activities and the gene expression. So cells are the basic functional units of the body. They are the basic functional unit of life. There are many cells that live on their own in the environment: bacteria, protistans, things like that. They come from a variety of shapes and sizes, and this diversity reflects their diverse functions. Cells are the oldest living life forms on the planet. They evolved, 3.9 billion years ago when the first bacteria evolved. I always say this is bacteria's planet. We just live on it. What you're seeing here is a typical cell, and this is going to be an animal cell because it doesn't have a cell wall, and it has centrioles, which animal cells are the only ones that are known to have these, and they are very important in mitosis, which we'll talk about at the end of the chapter. Here is a table that is table 3.1 in your book. It's blue colored in your current edition. And it goes through all the cellular components, their structure, and their function. And again, we're going to spend most of the time on the plasma membrane and the nucleus. The structure of the plasma membrane is phospholipid barrier between the intracellular and extracellular environments. The phospholipid has a hydrophobic center, and thus it's a double membrane. This double membrane with the hydrophobic center restricts the movement of water-soluble molecules, and ions. This is what makes it selectively permeable. Many substances are thus selectively allowed to pass through protein channels, and proteins and phospholipids are not trapped in the membrane, but they constantly move laterally. This is known as the fluid mosaic model. Phospholipids have a slightly oily consistency, and so they are moving around. Remember, cholesterol helps to stabilize the cell membrane because of that oily viscosity. Here we see the structure of the plasma membrane. This is figure 3.2 in your book, and it's that fluid mosaic model. It says the membrane consists of a double layer of phospholipids with the polar regions shown by the spheres oriented outward. So we've got the extracellular side and the intracellular side, and the hydrophilic polar regions are oriented towards those because they're a watery environment. And the nonpolar hydrocarbons with the wavy tails are oriented towards the center because they are hydrophobic and cannot interact with that watery environment. Proteins may completely or partially span the membrane. Carbohydrates are attached to the outer surface. And there's the cholesterol molecules that I just mentioned that are going to stabilize the cell membrane. Let's talk a little bit more about the membrane proteins. There's two principal types of membrane proteins: integral and peripheral. Integral proteins span the membrane, and peripheral proteins are embedded on just one side of the membrane. Protein functions are structural support, transport, enzymatic control of cell processes, receptors for hormones and other molecules, and the self markers for the immune system. There are other important components of the membrane, such as the glycocalyx and cholesterol. The glycocalyx is a system of glycoproteins, glycolipids, and polysaccharides that are attached to the outer plasma membrane. It serves as protection, gives the membrane shape, it has antigens on it, and it interacts with regulatory molecules and may even have possible signal transmission capabilities. Cholesterol gives flexibility to the membrane, and like I said, it helps to stabilize it because otherwise it would be this oily, viscous layer. The cholesterol kind of helps it become a little more waxy. Your next grouping of course outline, or the learning objectives is for the cell transport, and there are two principal types of cell transport: phagocytosis or phagocytosis and endocytosis. Phagocytosis is going to be, and this is the two principal types of the cell transporting substances into itself, is what I should have said earlier. My apologies. Then we'll see exocytosis, which is the transport out. So there's two principal methods that the cell will transport substances inside of itself: phagocytosis and endocytosis. All right, so the phagocytosis is the bulk transport of large extracellular substance into the cell, which is why it translates as cell eating. Some cells, like neutrophils and macrophages, can perform amoeboid movement by extending pseudopods to pull the cell forward. It relies on the bonding of proteins called integrins with extracellular proteins. And the pseudopods engulf bacteria, dead cells, or other organic materials, and then they fuse together to form a food vacuole. The food vacuole fuses with a lysosome, and then the material is digested. It's very important for body defense, inflammation, and apoptosis. So remember, the cell is the basic structural unit of life, and I had told you that it can live on its own, like amoebas or protistans, when they live like in the environment on their own. And so it's not surprising that we have amoeboid-like cells inside our own bodies that aid in our defenses. This is an illustration, figure 3.3, that says it's a colored scanning electron micrograph of phagocytosis. And the phagocytic tissue macrophage is engulfing tuberculosis bacteria, shown in pink. And it's the pseudopods that's engulfing it. The pseudopods will fuse so that the bacteria will be inside the cell within an enclosed vacuole. This is the coolest picture I've seen of the pseudopod engulfing the bacteria. I've always seen like images of the phagocyte going in and actually like engulfing the bacteria from the side, but I've never seen the 3D view of the actual pseudopod. So this is a really cool picture. First time I've seen it, so that's neat. Go book. All right. So then we have the endocytosis. As mentioned, this is the other principal process for bringing large materials into the cell. And within endocytosis, we see that the plasma membrane furrows inward rather than extending outward. That's a major difference. So it's not using the pseudopods. It's invaginating inward. So a small part of the membrane surrounding the substance pinches off and is brought in as a vesicle. Penocytosis is nonspecific. It's going to be found like in the small intestines. It's a way that nutrients are absorbed. Receptor-mediated endocytosis is very specific. It has receptor proteins in the membrane that will bind to the substance to be brought in. So the examples of that would be cholesterol going in the cell, the AIDS virus, and the hepatitis virus. This is figure 3.4. It's an electron micrograph showing endocytosis by a liver cell. The plasma membrane can be seen to invaginate and create a vesicle that pinches off, containing extracellular material. Invaginate just means to fold inward. And then there's exocytosis. This is how cells get materials out of their little bodies. And so large cellular products such as proteins and other molecules are moved out of the cell. The Golgi apparatus packages proteins into vesicles that fuse to the plasma membrane, and the contents spill out of the cell. Nerve endings use exocytosis to release their neurotransmitters. That's a good example. Hormones are also secreted by exocytosis. Many cells have cellular extensions. So we're moving on to those now, the cilia and the flagella. These are going to be extensions of the cytoskeleton. Cilia are going to be tiny hair-like structures composed of pairs of microtubules, which is part of the cytoskeleton, that project from the plasma membrane. The primary cilium, most cells have this non-modal cilium with a 9+0 structure. That's the way that the microtubules are arranged in the cilia, and this allows the sensing of fluid movement. Modal cilia will be in unison to move substances through hollow organs, and they're particularly found in the cells of the respiratory tract and uterine tubes. The respiratory tract will move the mucus, and the uterine tubes will move the ova as it is expelled from the ovary and moves down the uterine tubes towards the uterus. They have a 9+2 arrangement, and we'll see a picture of that in a moment. We'll go into some detail. But before we do that, the base of each cilium has a pair of centrioles that are perpendicular to each other and called the centrosome. One centriole is parallel to the cilium and called the basal body, and it is involved in pulling duplicated chromosomes apart in mitosis. And I mentioned before that this is only found in animal cells. Here's the image I was telling you about, cilia, as seen with the electron microscope. Figure A, 3.5a, is the scanning electron micrograph of cilia on the epithelium lining the trachea. So it looks like we're moving bacteria and mucus out of there. And then we've got the transmission electron microscope micrograph of a cross-section of cilia, showing the 9+2 arrangements of microtubules within each cilium. And it's kind of cool because there's a little motor protein in it called dynein that will be what moves the cilium. It's like little arms, dynein arms, and they can push the cilia, and then they release and push and release and push and release. Then we have the flagellum. The only human cell that has a flagellum is the sperm, and it is a single whip-like structure that can propel a cell forward. It also has the microtubules with a 9+2 arrangement, and it is very quickly moving and will have a lot of mitochondria in the area surrounding the flagellum, so that it can really be powered like think of it like a motor, because that sperm has to swim fast, otherwise the female's immune system recognizes it as foreign and can kill it before it gets to the ovum to fertilize it into a zygote. Next, we have cellular extensions that are not going to arise from the cytoskeleton. Instead, it arises from the plasma membrane. And those are called microvilli. And these are folds in the plasma membrane that increase the surface area for chemical reactions and rapid diffusion. So this will be found in the intestines and the kidney tubules. Very important in the intestines because it aids in the penocytosis, which is, as we said, the non-specific endocytosis, where we're absorbing nutrients into the bloodstream. Figure 3.6 shows microvilli in the small intestine. So this is one cell, and the microvilli are going to show, looks like two adjacent cells. Ooh, it's two cells. So this is the junctional complexes. So they are so tightly together that they look like one. Wow. These are great pictures in this book, y'all. I hope you are are paying attention and take advantage of them. These are images I've never seen before. And so the microvilli are seen in this colored electron micrograph, which shows two adjacent cells joined together by junctional complexes. And this is important because we do not want anything to leak out of our intestinal cells. You've heard of leaky gut. So we do not want to compromise these junctional complexes. All right, so we're going to quickly go through the cytoplasm and the organelles. The cytoplasm and the cytoskeleton make up the material within a cell, and it includes organelles, a fluid called cytosol, the cytoskeleton, and inclusions. Inclusions are stored chemical aggregates such as glycogen granules, melanin granules, and triglycerides. We'll talk about the cytoskeleton first, which is appropriate since we just talked about some extensions like the cilia and the flagella. So the cytoskeleton is the organized system of microtubules and microfilaments throughout the cytoplasm. The proteins of the cytoskeleton are mobile. They organize the intracellular environment and allow the movement of muscle cells and phagocytic cells. The cytoskeleton will form cell division spindle that pulls chromosomes apart in mitosis. They also serve as a railway system for vesicles and organelles to move along using molecular motors of myosin, kinesins, and dyneins. I'd already talked to you about the dynein arms, and the kinesins are too cool. They are what carry the vesicles throughout the cytoplasm, and they have little feet that they walk on, and they walk along the microtubule railway. And then myosin is going to be one of the motor proteins of the muscle fibers, and it is what moves the actin and slides it during muscle contraction. We always had a phrase when I was in school that said, on a cellular level, I'm really quite busy. So pairs of microtubules move materials in opposite directions. Image 3.7 shows an immunofluorescence micrograph showing the microtubules. In these fibroblast cells, the microtubules are green and the nuclei are blue. And then over here, in figure 3.8, we see the formation of the cytoskeleton by microtubules. The microtubules are also important in the motility or movement of the cell and the movement of materials within the cell. Lysosomes are organelles filled with digestive enzymes. They fuse with food vacuoles after a phagocytic cell engulfs a bacterium or dead cell. The primary lysosome contains digestive enzymes and is more acidic than the surrounding cytoplasm. A secondary lysosome contains the partially digested contents of the food vacuole or worn out organelles. Besides digesting bacteria, lysosomes are responsible for autophagy and apoptosis. Autophagy is the process of digesting viruses, worn out or damaged organelles, and proteins in the cell. It's a vacuole called an autophagosome. Apoptosis is programmed cell death. The lysosome spills its contents, killing the cell. And I know it sounds bad, but it's part of cell regulation. And so if there's like a mutated cell that should not go through mitosis, apoptosis is very important. Also, the reason you have individual fingers is because apoptosis occurs to eat away that webbing between your fingers when you're in utero. Next, we have peroxisomes. These contain enzymes specific to certain oxidative reactions. They're oxidases, that's the enzymes. They're found in most cells, but are the most numerous in the liver. They're important for the metabolism of amino acids and lipids. They even oxidize toxic molecules, such as alcohol. Enzymes are used to remove hydrogen from an organic molecule and transfer it to oxygen. forming hydrogen peroxide. And then it contains the enzyme catalase, which converts hydrogen peroxide into water and H2 and oxygen. And that's why peroxide will bubble when it is on your skin. Mitochondria, the famous mitochondria. These are the sites of energy production through aerobic cell respiration. We're going to talk a lot about them in the next couple of chapters. The structure of mitochondria is that they have an inner membrane and an external membrane separated by an intramembranous space. The inner membrane is folded into crista to increase surface area for reactions. The central area is a fluid and is called the matrix. This image shows the mitochondria, and it is figure 3.9. So stay tuned. We're going to learn all about her. She's got her own DNA. She can divide through binary fission. There's evidence that suggests that she was once a free-living bacterium. And I say she because generally she is who goes into cytokinesis, into the daughter cells, and meiosis, and all that good stuff. And so she is generally who's passed along to the developing fetus. There may be some new evidence coming out, but that's the general rule of thumb at this time. All right, so most cells have mitochondria, and there can be thousands of mitochondria in a single cell. Mitochondria can migrate around the cell and fuse together or reproduce by fission. So this is all the stuff that I was kind of blabbing on about. They have their own DNA. They're all derived from the mama. They are mutations of mitochondrial DNA may contribute to aging and disease. There are more than 150 mutations of mitochondrial DNA presently known to contribute to different human diseases. Mitochondria can produce superoxide radicals that can provoke an oxidative stress and produce reactive oxygen species. Mitochondrial DNA contains 37 genes that code for 13 proteins, 2 rRNAs, and 22 tRNAs. We'll learn about rRNA and tRNA shortly. Okay, so moving forward, before we do that though, if you are interested, in your book on page 58, it talks about how the mitochondrial DNA is more primitive, consisting of a circular, relatively small, double-stranded molecule, and also that this symbiosis may not always benefit the host. For example, mitochondria produce the superoxide radicals, etc. It also talks about Leber's hereditary neuropathy, malus, and some other diseases if you're interested in more information. Okay, now to the ribosomes. These are the protein factories of the cell. Messenger RNA takes genetic information to the ribosome so a protein can be assembled. That's going to be translation. It's very small, the ribosomes. They're made of 2 subunits of ribosomal RNA, which is the rRNA, and a protein. They are found free in the cytoplasm or associated with the rough endoplasmic reticulum. Ribosomes serve as enzymes called ribozymes that are needed for protein synthesis. Figure 3.10 shows a ribosome and that it's composed of 2 subunits. This is a model of the structure of a ribosome, showing the smaller, which is lighter, and larger or darker subunits. The space between the 2 subunits accommodates a molecule of transfer RNA. That's tRNA, and that's the RNA that is needed to bring amino acids to the growing polypeptide chain. Now the endoplasmic reticulum, or ER. There's 2 principal types, but all in all, the ER is a system of membranous passageways from the nuclear membrane to the plasma membrane. The rough ER has ribosomes embedded on the outer surface. It is the site of protein synthesis for those transported proteins to the Golgi complex. And then the smooth ER has many functions depending on the cell. So it can detoxify, drugs and alcohol. It can help in the synthesis of lipids and the storage of lipids. It can also store calcium ions, like in the muscle tissues. Figure 3.11, pictured here, is the endoplasmic reticulum. A shows an electron micrograph of a rough endoplasmic reticulum. And the rough endoplasmic reticulum has B ribosomes attached to the surface, whereas the smooth endoplasmic reticulum, which is C, lacks ribosomes. The Golgi complex, aka Golgi apparatus, consists of stacks of hollow flattened sacs, and the cavities are called cisternae. One side receives proteins from the endoplasmic reticulum. These are packaged into vesicles called endosomes that bud off to fuse with the plasma membrane for exocytosis, or they become lysosomes. Proteins are modified within the cisternae based on the type of protein, and retrograde transport occurs when extracellular proteins brought in by endocytosis and then transported through the Golgi complex go to the endoplasmic reticulum. Figure 3.12 shows the Golgi complex. A is an electron micrograph of a Golgi complex. Notice the formation of vesicles at the ends of some of the flattened sacs. B is an illustration of the processing of proteins by the granular endoplasmic reticulum and the Golgi complex. And generally it shows a few of the pathways that they can go to. And the same is true here. If you look, we've got a secretory pathway. And that's going to be secretion of neurotransmitters or hormones or any other thing. And then here we see the lysosomes is a pathway. And we learned that there are two kinds of lysosomes, a primary lysosome for digestive enzymes and a secondary lysosome for helping to break down old organelles and cellular bits. All right, so now we're on to gene expression. This is a big part of the chapter and will lead to the next couple of chapters. So let's focus on this. A lot of your study guide questions and learning objectives come from this part of the chapter. Most cells have one nucleus. However, muscle cells have hundreds and mature red blood cells have none. So there's always exceptions. Oh, sorry, I was moving my... All right, the nucleus is enclosed by the nuclear envelope made of two membranes. The outer membrane is continuous with the rough endoplasmic reticulum, and the inner membrane often fused to the outer by nuclear pore complexes, which allow small molecules and RNA to move into or out of the nucleus through those pores. Figure 3.13 shows the nuclear membrane and the pores. A diagram showing the inner and outer nuclear membrane and the nuclear pore complexes. The nucleolus within the nucleus is also shown. And we'll see that the nucleolus is where ribosomes are synthesized. So DNA and genes. The nucleus contains DNA. A gene is a length of DNA that codes for a specific protein. The gene on the DNA is transcribed as messenger RNA, which can leave the cell. This is called genetic transcription. And so to scribe or write a script is to write something down. And that's what we're doing here. We're writing the gene of the DNA onto a molecule of RNA. And the reason we do this is because the RNA can leave the nucleus, whereas the DNA cannot. And so once the messenger RNA leaves the nucleus, it's translated at the ribosome to assemble the proper amino acid sequence. And this is called genetic translation. And to translate is to, take one language into another. And so we're taking the language of DNA, the gene, We've transcribed it, written it onto a messenger RNA, and then we've translated that message on a ribosome, and we've translated it into a sequence of amino acids. And so these two steps of transcription and translation can be called genetic expression. Here's the nucleoli. The nucleus also has one or more darker regions not surrounded by a membrane. These are called nucleoli. The nucleoli contain the DNA that codes for the production of ribosomal RNA. The genome and the proteome. The genome is all the genes in a particular individual or all the genes in a particular species. The Human Genome Project has found that humans have about 20,000 protein coding genes, which makes up about 1% of the genome. Most genes code for various types of RNA. Some parts serve as promoter regions, some parts are enhancer regions, and more than half consists of jumping genes. It says that jumping genes can change their locations. Many of these are retrotransposons, where the DNA involved is copied into complementary RNA. This is then used to synthesize a DNA copy that is inserted into a different location in the genome. And the performance of these and other functions, most of the genome that lies outside of the historical definition of genes, the protein coding regions, is transcribed into RNA at some time. Until recently, it was believed that one gene could be defined as coding for one polypeptide chain. However, each cell produces over 100,000 different proteins, so the number of proteins greatly exceeds the number of genes. The term proteome has been coined to refer to all of the proteins produced by the genome. This concept is complicated because in a given cell, some portion of the genome is inactive. There are proteins produced by a neuron that are not produced by a liver cell, and vice versa. Further, a given cell will produce different proteins at different times as a result of signaling by hormones and other regulators. So, how does a gene produce more than one protein? This is not completely understood, but part of the answer may include the following. So, an mRNA is altered after transcription by cutting and splicing in different ways. This is called alternative splicing. Or proteins are made of many polypeptide chains that can associate in different combinations. Or protein modification can occur by adding a lipid or a carbohydrate. And there is also a great variety of post-translational modifications of proteins that are made after the polypeptide chains have formed. And these include methylation and phosphorylation. Now let's talk about chromatin. DNA in the nucleus is packaged with proteins called histones to form chromatin. Histones are positively charged and will interact with negatively charged DNA to cause spooling, and this creates particles called nucleosomes. Euchromatin is active in transcription, it's extended, it has chemical changes in histones such as acetylation, and it allows for molecules access to the DNA during gene expression. Heterochromatin is inactive regions, highly condensed, and it occurs where much of the DNA is inactive. As long as an area of chromatin is in the condensed state, its genes are inactive. In the euchromatin, genes may be activated or repressed at different times. The figure 3.14 shows the structure of chromatin, and part of the DNA is wound around complexes of histone proteins, forming particles called nucleosomes. And these histones are very important because they're different between the different domains, like the archaeans, the bacteria, and the eukaryotes. And so our histones are going to be different than bacterial histones, and the mitochondria will have histones that are more like a bacteria. Hence, remember the endosymbiotic theory mentioned earlier, where mitochondria existed particularly as a bacteria. All right, this is figure 3.15, and it shows how chromatin structure affects gene expression. I keep seeing macaroons. Just want a macaroon cookie now. Okay. All right, so the first one is showing the condensed chromatin, where nucleosomes are compacted. And then the second, is the acetylation of chromatin, and it produces a more open structure. Then we have transcription factors that come in. They attach to chromatin and activate genes, producing RNA. And then finally, deacetylation causes the compaction of the chromatin, silencing genetic transcription. Gosh, these chemical processes are just so amazing. Like I said, cell biology is what won me over to biology as a whole. So the figure text states that the ability of DNA to be transcribed into messenger RNA is affected by the structure of chromatin. So the genes are silenced when the chromatin is condensed. Acetylation, which is the addition of two carbon groups, produces a more open chromatin structure that can be activated by transcription factors producing mRNA. Deacetylation is the removal of the acetyl groups, and it silences genetic transcription. All right. Oh, sorry. Turning page. RNA synthesis is also called transcription, and it involves start and stop regions at the beginning and end of the gene. It involves promoters, which are areas of DNA that are not part of the gene but tell enzymes involved where to begin. And then transcription factors that bind to the promoter to begin transcription. It says each gene is a stretch of DNA that is several thousand nucleotide pairs long. The DNA in a human cell contains over 3 billion base pairs, enough to code for at least 3 million proteins. Because the average human cell contains fewer proteins than this, 30,000 to 150,000 different proteins, it follows that only a fraction of the DNA in each cell is used to code for proteins. And as we just saw, some of the DNA may be inactive, or it could even be redundant, and some serves to regulate those regions that code for protein. In order for the genetic code to be translated into the synthesis of specific proteins, the DNA code first must be copied onto a strand of RNA. This is accomplished by DNA-directed RNA synthesis, which is the process of genetic transcription. All right, so let's see. We started with the transcription and involving the stop and start region, promoters, and transcription factors. And so next, we're going to talk about RNA polymerase. This is an enzyme with the ACE there and polymer. It's building a polymer. So RNA polymerase will break the hydrogen bonds between the base pairs of DNA, and it assembles the appropriate RNA nucleotide. RNA nucleotides will pair up to the DNA template, and there is a complementarity base pairing that is used where A will bind to T, C will bind to G, G will bind to C, and T will bind to U. A stands for adenine, T stands for thymine, G stands for cytosine, or C stands for cytosine, G stands for guanine, and U stands for uracil. So RNA will have uracil instead of thymine. RNA synthesis will also form the precursor messenger RNA, mRNA, that detaches from the DNA template, and only one free DNA strand is transcribed. Okay, so once we have built our beautiful RNA, then of course it can go off into the cytoplasm to be translated. So now there's more than messenger RNA though that's being built. There's going to be transfer RNA and also ribosomal RNA. But let's go back to the mRNA first, and we'll even look that there's a precursor messenger RNA, a pre-mRNA, and this is going to be directly made by transcription. I love the way this book is going to distinguish between the two, because it is neat. There is, you know, some modification that the precursor mRNA has to go through before it can leave the cell. It has to have like these little caps put on it and all kinds of cute things. So once the messenger RNA is made from that modified pre-mRNA, it's got little caps that are put on it. It cuts out any like non-coding section through a process called splicing, and we make an actual code that will make a specific protein, and that's the mRNA that can then leave the nucleus and go into the cytoplasm. And figure 3.17 is going to show the process of pre-mRNA into mRNA. So stay tuned. And then transfer RNA is going to carry amino acids to mRNA for translation. It's got little area on it that will attract the particular amino acid, and then it's going to have what's called an anticodon on it that will have complementary base pairing to the mRNA, and it can then add the amino acid in that way. And then there'll be peptide bonds formed between the growing polypeptide chain. It's a beautiful process. Then we have ribosomal RNA, rRNA, and this, along with protein, forms the ribosomes, which are the site of translation. They can also act as the enzyme. So lots of RNA, and I think I had mentioned earlier, I think even in the last chapter with the chemistry, that RNA is older than DNA, and it was thought to be like the first hereditary molecule on the planet, followed by DNA as cells became more complex. All right, so now, as promised, pre-mRNA modification. So the precursor messenger RNA is altered in the nucleus before it leaves as the messenger RNA. Protein or portions of the gene that do not actually code for proteins are called introns, and they must be spliced out. Introns may regulate the expression of the area that do code for proteins, and they generally within the pre-mRNA, and they're not on the ends. We put the little caps on the ends. So the portions that are kept are called exons because they are expressed. Expresson. And then alternative splicing allows one precursor mRNA to be used to make multiple proteins. And exons are joined together by spliceosomes and SNRPs, which are small nuclear ribonucleoproteins. Figure 3.16 shows RNA synthesis via the process of transcription. So you can see we're building the single-stranded RNA off of the DNA. And I know that's the RNA right away simply because it has uracil rather than thymine. So the uracil will bond to the adenine. Guanine binds to cytosine. We see a uracil going to bond to the adenine. Cytosine to the guanine. Uracil, adenine, cytosine to guanine. And then here we see an adenine in the DNA because we're twisted now, the helix. So again, a uracil to the adenine, a guanine to the cytosine, a cytosine to a guanine, and we see an A down here on the RNA, so we know that that would be binding to a thymine on the DNA. And then figure 3.17 shows the processing of pre-mRNA into mRNA. Non-coding regions of the genes called introns are going to produce excess bases within the pre-mRNA, and these excess bases are removed, and the coding regions of mRNA are spliced together. Exons can be spliced together in different sequences to produce different mRNAs and thus different proteins. Then we put on a little cap on each end, and we can leave the nucleus and go off to translation. Now we're talking about RNA interface, RNAi. Turn the page here to where it talks about that, page 66. So RNA molecules that don't code for proteins may prevent some mRNA molecules from being translated. 3 types. There's siRNA, short interfacing, miRNA, microinterfering, and circRNA, circular RNA. And then it goes on to talk about those on page 66. Says the short interfering RNA is a double-stranded RNA. It's pretty fantastic. Then we've got microRNA is going to enter a particle called the RNA-induced silencing complex. It's a single-stranded, and it binds to complementary base pairings, and it suppresses translation of the gene. Then we have circular RNA, and it says it appears to be thousands of these formed by splicing through head-to-tail covalent bonding of exons from the same pre-mRNA. And it can serve as a sponge to prevent microRNA molecules from suppressing the translation of their target mRNA molecules. They inhibit the inhibitory microRNA. So because the functions of microRNA and short interfering RNA have been more thoroughly studied, however, the rest of this section is devoted to them. All right, so here we go. Short interfacing RNA, cRNA, or siRNA. So it's formed from a larger double-stranded RNA molecule that leaves the nucleus. An enzyme in the cytoplasm called dicer cuts the strand into short segments 21 to 25 nucleotides long, and it's a mechanism used to fight viral infections. Ooh, I wonder if that's where the COVID vaccine had come from. Interesting. Then we have the microRNA, or miRNA, formed from a single-stranded RNA that doubles over in hairpin loops of double-stranded RNA. It's processed in the nucleus by enzymes and by the dicer in the cytoplasm into short 22 nucleotide double-stranded segments. It enters a particle called the RNA-induced silencing complex, or RISC, that binds to mRNA to suppress translation. And this is also interesting to me because when I teach, the evolution of RNA in my General Biology 2 class, we talk about how RNA can form hairpin loops and do all kinds of very interesting things to, express genes, or as we're seeing, inhibit gene expression. So here's the circRNA, the circular RNA, and just as I had mentioned earlier from the book, it says there appears to be thousands of these formed by the splicing through head-to-tail covalent bonding of exons from the same pre-mRNA. So that's pretty much verbatim from the book. And if we remember, they are going to inhibit the inhibitory microRNA. Okay, so in summary, one of the strands of siRNA and miRNA enter a protein particle called the RNA-induced silencing complex, or RISC. These can combine mRNA where it is complementary, causes destruction of the mRNA so translation is prevented, the gene is silenced. Scientists estimate that at least 30% of the human genes are regulated by microRNA. So looks like short interfering RNA is going to be important in, as we saw, fighting viruses. We saw that microRNA is going to silence gene expression by destroying the messenger RNA. And then we have learned that circular RNA is going to inhibit microRNA so that gene expression continues to occur. It says that scientists estimate there are 700 to 1,000 different microRNA molecules in the human genome. And as we saw here, that's 30%. Separate genes code for many of these, but others are derived from introns within genes that code for proteins. In that case, when pre-mRNA is cut and the exons are spliced together to make an mRNA, an intron removed in the process is then processed into a microRNA that regulates the mRNA. Oh my gosh. So we're just learning about all this stuff, and there are scientists out here who actually saw this, discovered this, could name this, and understand what in the world was going on. Wow. All right, so it says one microRNA can regulate the expression of more than one messenger RNA. And this is possible because one microRNA can be incompletely complementary to a number of different messenger RNA molecules from different genes, causing them to be silent. So it doesn't even have to bind to the entire mRNA molecule. So in this way, a single microRNA may silence as many as an estimated 200 different mRNA molecules. Wow. I love this stuff. I hope you do too. I know you're like, oh my gosh, but it's also fascinating and complex, beautifully complex. The body is an incredible biological machine. And again, these things too aren't just in the human body. We're not the only things made up of living cells. A lot of this stuff is bacterial even. And so if it's bacterial, it will be in the protists, like amoebas. It'll be in plants, and animals, and fungi, all this awesome stuff. All right, so moving on. Now we're going to talk about what's called CRISPR. So it says the use of RNA interface as a technique to experimentally silence genes has been tempered by the introduction of a new technology that has generated much excitement. This employs a system called CRISPR-Cas9, derived from a bacterial immune mechanism that helps bacteria defend against viruses by cutting the viral DNA. CRISPR is an acronym for Clustered Regularly Interspersed Short Palindromic Repeats. Yeah, we'll just call it CRISPR. It's a unique sequence of bacterial DNA on either side of some target gene of interest to the scientists. The CRISPR target gene enlists the function of nucleus enzyme Cas9, which cuts DNA, and an RNA molecule that guides the Cas9 to the specific gene on the target chromosome. Excuse me. Scientists can use the CRISPR-Cas9 to remove specific genes. By changing the CRISPR-Cas9 system in certain ways, they can also inactivate genes without removing them. This technique is more specific than gene inactivation through RNA inhibition using microRNA. In a twist on this theme, CRISPR-Cas9 has also been used to activate instead of repress specific genes. The book ends up by saying this powerful tool for manipulating the genome holds great promise for biomedical research and future medical applications, but it does have the potential for ethical abuse. Wowzers. Okay, well, if any of you are going into genetics, you can play with a lot of this stuff. But for now, let's move on to Chapter 3, Section 4, which is going to be on protein synthesis and secretion. And then the last part of the chapter is going to be 3.5, which is then going to go over DNA synthesis and cell division. We finally end with meiosis, which is included in 3.5. All right, so moving on. Protein synthesis and secretion. So this should be easy for you after all that RNA interference stuff with the short interfering RNA, the microRNA, the circular RNA, the CRISPR. So protein synthesis at this point, you're like, oh yeah, okay, it's called genetic translation. I already know about this. mRNA was transcribed in the nucleus. Now it's out in the cytoplasm where it's going to be translated, if it's not silenced, as we know. And it's going to go to a ribosome. So mRNA passes through a string of ribosomes to form a polyribosome. A group of 3 bases on DNA called a triplet gives the complementary 3-base sequence in mRNA called a codon. And I've already mentioned to you that the tRNA is going to have an anticodon, which will then bond to the codon, attach the amino acid to the growing polypeptide chain. That's the gist of it all. So the codon codes for an amino acid, So the order of the codons gives the order of the amino acids that will be formed in the polypeptide. Figure 3.18 shows the rendering of a polyribosome. A strand of mRNA runs through the ribosomes. As translation occurs, polypeptide chains emerge from the ribosomes. And this is so great because it's like a protein factory, and that's exactly what cells are. So look at all of the ribosomes that are working on this particular mRNA. And off of that, we are going to have the newly synthesized proteins growing, and it's going to be a ton of protein. So that makes it super efficient. All right, Henry Ford of the cell, that factory. All right, so we see here in table 3.2 triplets, codons, and amino acids. And this is, of course, in blue in your book, and it's in that peach colored here, but it all says the same thing. So we see that the original DNA triplet that's still in the nucleus was TAC. And so to build the mRNA codon off of it, we are going to do that complementary base pair. And so we know that A will bond to T, and RNA has that. That's fine. T is going to bond to A, but RNA doesn't have that. It's got uracil instead. So we're going to put uracil bonding to A. And then we know that guanine, or G, will bind to cytosine. So the RNA codon is AUG. It's a complementary base pair. And so the amino acid that it codes for is the methionine. So once the transfer RNA comes in with the anticodon, you may have guessed that the anticodon is going to read U, A, C. Okay, so this is figure 3.19. It's a big figure. It takes about half of a page. And it shows transcription and translation. So the genetic code is first transcribed into base triplets or codons in mRNA, and then they're translated into specific sequences of amino acids in the polypeptide. So basically what's happening is this up here, the DNA triplet strand, this is where the triplets are. So TAC is the DNA triplet. Then when we look at the messenger RNA, that's the codon. And so remember, we're using complementarity base pairing. So if the DNA says T, then the mRNA says A. If the DNA says A, the mRNA says U, because we don't have T. If the DNA says C, then the mRNA says G. And then we have to go into the ribosome, and a transfer RNA is going to come in with an anticodon, which we haven't even talked about yet. So when we get there, you're going to be like, all right, anticodon. Anticodon is then going to bind to the messenger RNA, and then it will join the amino acid to the growing polypeptide chain. So let's see what the anticodon would say. If the messenger RNA says A, then the anticodon would say U. If the messenger RNA says U, the anticodon will say A. If the messenger RNA says G, the anticodon will say C. So that's going to be U, A, C. Ta-da! So here's transfer RNA. It's a single strand of RNA bent into a cloverleaf shape that is further twisted into an upside-down L shape. It's pretty fabulous. Again, going way back to early life on this earth, RNA is where it's at, I tell you. All right, so one end has the anticodon, which is 3 nucleotides that will be complementary to the proper codon on the mRNA. The other end has the appropriate amino acid bonded to tRNA by amino acyl tRNA synthetase enzyme. Blah, blah, blah. So tRNA with a given anticodon can only bond to one specific amino acid. There are 61 different codons for the 20 amino acids, so there are different tRNAs and synthetase enzymes specific for each codon and amino acid. Figure 3.20 shows the structure of the transfer RNA, and it's that simplified cloverleaf representation and the 3-dimensional representation. And you can see the amino acid accepting end, and it's very specific. It can only accept that particular amino acid. So all these things have to be in place in order for your proteins to be properly transcribed and translated. So we have to be able to correctly transcribe the mRNA from the DNA. We have to correctly process it with sniping out the introns and splicing together the exons and putting little caps on them. And then the mRNA has to be able to leave the nucleus. It has to then be able to interact with all those crazy RNA interference molecules, the siRNA, the microRNA, the circular RNA, all that crazy stuff. Then, once it gets to a ribosome, it's got to then have the appropriate transfer RNAs with the appropriate amino acids to come in and code for these proteins and join the correct amino acids to the growing polypeptide sequence. Wow. Again, it just blows my mind. This is my absolute favoriteest stuff because this is the stuff of life. Like, this is the stuff that's been worked on for billions of years and has ultimately given us the diversity of life on this planet. And, you know, whatever your creation origin stories, etc., I always tell students, you know, even if you are religious and have, you know, the creator in mind, I always tell students this is like the media with which the creator works. If you don't subscribe to that, then, you know, you can understand that given billions of years of time, you know, and chance and natural selection, this stuff can form. If you like to think of it in both ways, that the creator put this stuff in play and then evolution took over and has rolled with it, it's just, it's beautiful. I love it so very much. And I'm sorry to go off on a tangent, but I just want to emphasize to you the importance of this and how it's all accumulated into us, the human being. And, you know, with you studying medicine and going into all of this, treating all of these diseases and disorders and things like that, you know, it's amazing that anything even goes right, isn't it? So anyway, just every day that you feel good and are healthy and you're making your proteins correctly and, you know, all of these things are going right for you, that's a good day. And it's a day to be blessed. And I hope that as you learn this stuff too, you really just love your body for what it's doing for you. And you could step outside of your, you know, insecurities about maybe the way you look or anything like that. And you can focus inward on just the beauty of your machine. So anyway, there's that. And I won't go off on many other tangents, but this is the stuff that just really gets me. And like I said, this is the stuff of life. And we'll see if I leave this in the video or, no pun intended, splice it out. But without further ado, this is the next part of the section 3.4, the protein synthesis and secretion. So here we're at the actual formation of the polypeptide. And it's basically what I've just said over and over again. So you've probably already read this as I was blabbing on. But you know that at this point, the mRNA, you know, God willing, has gotten to the ribosome. It's moving through the ribosome. The proper transfer RNA is attaching at each codon with its anticodon. The amino acids attached to the transfer RNAs form peptide bonds to each other, and they disassociate from the tRNA and go off to bind with another amino acid. And this continues until a stop codon is encountered and the whole complex dissociates. Interaction between amino acids of the growing polypeptide chain causes bends and folds into secondary and tertiary structures. So we're not even done yet. We're making disulfide bridges. We've got positive and negative interactions. We've got polar and nonpolar. We've got hydrogen bonds forming. It's just wild. And there's even chaperone proteins that aid in the formation of correct interactions and folds. Like I said, on a cellular level, you're so busy. And that's why we have to eat and get our energy. And that's what we're going to talk about in the next couple of chapters. Because we got to power these machines and we do it through our food. So in the discussion of week 3, I ask you about the foods you eat, and this is why. Because this is where it goes. It goes to power all of these cellular processes. And I know that teaching this material helps me make better decisions on what I eat. Every now and again, of course, I'll cheat and I'll eat something that's really crap for me. But mainly I try to focus on things that's going to feed these beautiful molecules and help my protein synthesis and help my body to do what it needs to do to keep me alive and healthy. And before I risk getting off on another tangent, let's take a look at what this figure says. It's figure 3.21, and it's showing the ribosome. And so we see over here, number 1, we've got the anticodon of the tRNA bonding with the codon of the mRNA so that a specific amino acid it carries can form a peptide bond with the last amino acid of a growing polypeptide. And then number 2, the tRNA that has brought the next to last amino acid disassociates from the mRNA so that the growing polypeptide is attached only to the last tRNA. And then in number 3, another tRNA carrying another amino acid will bond to the next codon in the mRNA so that this amino acid will be at the new growing end of the polypeptide. So newly formed proteins destined to leave the cell are made on the rough endoplasmic reticulum. The first approximately 30 amino acids are hydrophobic and are attracted to the lipid portion of the membrane of the rough endoplasmic reticulum. The growing polypeptide chain enters the cisternae, or the internal chambers of the endoplasmic reticulum. The hydrophobic leader sequence is removed, and other portions may be removed or added. For example, the conversion of pre-proinsulin to insulin, the middle region is removed and the ends are bonded together. And that's shown here in figure 3.22, how secretory proteins enter the endoplasmic reticulum. So of course they're going to have a hydrophobic end that's able to go through the lipid portion of the membrane. It's just so brilliant. The protein destined for secretion begins with the leader sequence that enables it to be inserted into the cisternae, or the cavity, of the endoplasmic reticulum. Once it has been inserted, the leader sequence is removed and carbohydrate is added to the protein. This is what the conversion of proinsulin into insulin looks like. So the long polypeptide chain called proinsulin is converted into the active hormone insulin by enzymatic removal of a length of amino acids shown in green. The insulin molecule produced in this way consists of 2 polypeptide chains, which are the red circles, joined by disulfide bonds. Because proteins have so many important functions, the process of genetic transcription and translation have to be physiologically regulated. All right. So now we're going to see that secretory proteins are next sent to the Golgi complex. The proteins may be further modified, including the production of glycoproteins. Proteins are separated according to their function and their destination, and the proteins are packaged and shipped in vesicles to their destinations. So we often talk about the Golgi complex as a post office or like a UPS center. And so that's what they're doing. They are separating, packaging, and shipping the proteins. And remember, the vesicles aren't just floating through the cytoplasm of the cell. They are being walked. They're like the postman or woman or the UPS driver. And these motor proteins, these kinesins, are literally carrying the vesicles across the microtubule railway towards the plasma membrane so that they can be secreted by exocytosis. Protein degradation. Regulatory proteins are rapidly degraded, making their effect short-lived. This allows for greater control of cell functions. So here we're going to see proteases. These are enzymes that are in the lysosomes, and they digest proteins. Proteins outside the lysosomes are tagged by a molecule called ubiquitin, which marks them for degradation by a proteasome. Ubiquitin may also tag membrane proteins and organelles for destruction. And remember, we learned that there's that secondary lysosome that will do that. It says degradation of ubiquitin tagged proteins within proteasomes eliminates defective proteins. For instance, incorrectly folded proteins produced in the endoplasmic reticulum, and it promotes cell regulation. For example, the stepwise progression through the cell cycle requires the stepwise degradation of particular cyclin proteins. I need to take a nap after this. You might as well. Let my cells rest and make their proteins and their mRNAs and their rRNAs and their tRNAs and their polypeptide sequences and all of this good stuff. So not only is all that going on inside the cell just to maintain the health of the cell and thus your body, but cells are also undergoing DNA synthesis and cell division when they need to, because your hair is always growing, your nails are always growing, your nose is growing, everybody's nose gets bigger, your ears get bigger, right? Your skin is always regenerating, your bones are always regenerating. So not only are cells constantly, synthesizing proteins and modifying proteins, but they are also synthesizing DNA for cell division. Again, that's why the next couple of chapters we got to talk about the foods we eat and the water we drink so that we can power all of these incredible processes in our body. All right, so now we're in section 3.5 on DNA synthesis and cell replication. So let's start with DNA replication. Before cell division can even occur, each DNA molecule must replicate itself so that one of each copy can be distributed to two new cells. My dad was a doctor when he was alive, and then had to retire, and then he went into teaching, and he taught anatomy and physiology. And he told me that if you take the DNA out of one cell, it could stretch to the moon or around the world a couple of times. And so we've got to duplicate all that DNA in order to distribute it into two new cells. Again, I'm not going to go crazy with any digressions. I've done my part. I've talked about how amazing this stuff is. So let's go on. DNA replication involves several types of enzymes. Helicases break hydrogen bonds between the DNA strands. So helicases, it's a double helix, right? The DNA. ACE means enzyme, so the helicases break the double helix, and this creates a fork in the double-stranded molecule where nucleotides can be added to both ends. DNA polymerases then bind the complementary nucleotides to each other, forming new complementary polynucleotide chains. And here, since we're building DNA, we're not going to have uracil in the mix. It's going to just stick with the A and T, G and C. So two new molecules are being made from the original one, each with half old and half new DNA. This is called semi-conservative replication. Which you do have a question on that in your study guide with the learning outcomes and all that. And so here, figure 3.22, another big figure that takes up a good half of the page or more, and it shows the replication of DNA. So each new double helix is composed of one old and one new strand. The base sequence of each of the new molecules is identical to that of the parent DNA because of its complementary base pairing. And so we see the region of the parental DNA helix, both backbones are light colored. And then we see the region of replication. The parental DNA is unzipped by the helicase, and new nucleotides are pairing with those in the parental strands. So then the region of completed replication, each double helix is composed of an old parental strand, light purple, and a new daughter strand in blue. I don't know about you, but I don't see blue anyway. I see dark purple and light purple. So the light purple is the old strand, and this dark purple is the new strand. And the two DNA molecules formed are identical to the original DNA helix and to one another. Now, can there be mistakes in any of these processes that we've talked about so far? Absolutely. And of course, some of these mistakes, which of course are called mutations, can be harmful. Some can be beneficial, and some can be neutral. But over the billions of years of life on this planet, ultimately it's given us rise to all the diversity that we have of life on this planet. But I'm going to stick with the information here and not go off on my tangent again. And we're going to talk about the cell cycle. So, introduction to the cell cycle is divided into interphase, mitosis, and cytokinesis. Interphase itself, you might have heard of as cell resting. It's not resting. We've learned that the cell is doing a lot of stuff throughout the day, every day. So when it's in G1, the G1 phase, G stands for gap, but it's not really gapping nothing. It's performing the physiological functions characteristic of the tissue in which they are found. So all those protein synthesis and modifications and stuff are going on here. Nutrient exchange, oxygen exchange, all that good stuff. Secretion of substances, metabolic waste products, all that are happening in this G1 stage. And then the S phase is not going to occur until the cell is going to divide. So if a cell is going to divide, it replicates its DNA in a part of interphase known as S, which stands for synthesis. Once DNA is replicated in the S phase, then it's going to go into the G2 phase, where chromatin will condense to form the thick structures that you know of as chromosomes. Though condensed, the chromosomes are not yet in their more familiar visible form. And they will not do that until the first stage of mitosis. So mitosis is going to be cell division, and it's nuclear division. And so mitosis is divided into prophase, metaphase, anaphase, and telophase. So this is when the nucleus is actually going to divide. And then if cytokinesis occurs and the cells, divide into two daughters, that is going to overlap the last parts of mitosis, and we have made two new daughter cells. If cytokinesis doesn't occur, then we just end up getting a large multinucleated cell called a syncytium, and that can happen too. Stay tuned. Okay, so in the cell cycle, this is figure 3.25, the life cycle of a cell. The different stages of mitotic division are showed. It should be noted, however, that not all cells undergo mitosis. Some are going to be stasis in G1, and they're going to do all their amazing, cellular processes, and they'll never divide into two daughter cells. But those that do will then go into the S phase, and then once they're in the S phase, they're committed to the G2 phase and then mitosis. Regardless, interphase makes up the bulk of the cell cycle. Mitosis usually occurs over a few hours. Okay, so in interphase, lots of RNA synthesis is occurring. Here's the G1 gap phase. The cell is performing the functions characteristics of cells in that tissue. And then you've been looking at this part in your book called the cyclins in P53. Cyclin D moves the cell quickly through G1. Overactivity of the gene for cyclin D has been implicated in some cancers, and those are called oncogenes. So P53 is a transcription factor that can stall a gene in the G1S checkpoint, repair DNA damage, or promote apoptosis, which will kill the cell, and that's a tumor suppressor gene. So the P53 is a good gene. It's the mutations in the P53, I believe, that is what can lead to cancer. And that's what you get tested for, like with breast cancers, is that mutation. So it's not working. That means that you have risk for developing cancer. So if a cell does not divide, it remains in a modified G1 phase its whole life. So DNA damage or errors in DNA replication can produce mutations. Mutations can also be produced by the insertion of lengths of DNA by viruses or by retrotransposons into the chromosome. Over the course of time, successive mutations in somatic cells can cause cancer and are also believed to contribute to aging. Cancer occurs when mutations allow a cell to escape normal restraints on cell division and proliferate to produce a clone of abnormal cells. So again, that's why we need this tumor suppressor gene known as p53. And it refers to the protein coded by the gene, which has a molecular weight of 53,000. And so, as we saw, it's a transcription factor. It can bind to DNA and activate or repress a large number of genes. So it says a normal p53 gene protects against cancer caused by damage to DNA through radiation, toxic chemicals, or other cellular stresses. The ability of p53 to suppress cancer by causing cell cycle arrest or apoptosis is reduced in over 50% of all cancers. In about half of these cases, the gene for p53 is mutated, and that's by a point mutation, which causes a change in a single amino acid in the p53 protein. In the other half of all cancers associated with inadequate p53 action, the p53 is normal, but its ability to function is reduced. This can occur if there's defects in other proteins needed for the p53 to function. Scientists are currently investigating molecules that might restore p53 activity as potential treatments for cancer. All right. So moving forward, as mentioned earlier, in the S phase, if the cell is going to divide, it performs DNA replication at this time. And then in the G2 phase, chromosomes start to condense. They consist of two strands called sister chromatids joined by a centromere. And once they get fully into mitosis, they will form the chromosomes that you know and love. So it's duplicated at this point. So we've already synthesized our DNA. So these are called sister chromatids. So they're two strands of identical DNA, and they're ultimately what's going to be separated during mitosis. So figure 3.26 shows the structure of chromosome after DNA replication. At this stage, a chromosome consists of two identical strands called chromatids, sister chromatids. Prior to this stage, when the DNA is in a more extended and less visible form, each chromosome consists of only one copy of the double-stranded DNA. So that's not this. This is when it's already been duplicated. All right, cell death, necrosis. The cell dies pathologically. We don't like that. Due to deprivation of blood supply. That's disease, necrotic wounds, bed ulcers, scary stuff. And then apoptosis may be triggered by extracellular molecules known as death ligands. And then they also can be, all cases they're going to be from the capsaicin. So it says here, in all cases of apoptosis, a group of previously inactive cytoplasmic enzymes called capsaicin. I want to say capsaicin, but it's caspasis. I'm going to try to think of Casper, the friendly ghost. Only this time, they might be friendly because apoptosis is going to really help us to trigger death due to DNA damage, cancer, infection, or oxidative stress. So caspases are friendly, and they're going to help protect us from damaged cells that could otherwise lead to disease. So Casper, the friendly ghost, caspases, the friendly enzymes that make our cells that are sick and otherwise dangerous into ghosts themselves. All right, so a cell marked for death releases chemicals that attract phagocytic macrophages that engulf the cell and digest it with its lysosomes. Knocked out mice with their p53 gene removed are used to study cancer and apoptosis, and they talk about that in the book in both sections of the p53 and then cell death. And it says that using mice with their gene for p53 knocked out, scientists have learned that p53 is needed for apoptosis that occurs when a cell's DNA is damaged. DNA damage occurs in response to ultraviolet light in cells exposed to sunlight, all forms of tobacco, cancer-causing chemicals, including those in foods, such as heterocyclic amines and overcooked meats, not to mention all the other crap we put in our preserved foods, and ionizing radiation. There's a whole lot more that can mutate DNA, but that's just a good little group of examples. The damaged DNA, if not repaired, activates P53, which in turn causes the cell to be destroyed. If the P53 gene has mutated to an ineffective form, however, the cell will not be destroyed by apoptosis as it should be. Instead, it will divide to produce daughter cells with damaged DNA. This may be one mechanism responsible for the development of the big C cancer. Okay, so now let's go on to mitosis. And this takes me back. I first started learning this in mid-school. And we've got prophase, metaphase, anaphase, and telophase. So, at the end of the G2 phase of the cell cycle, which is generally shorter than G1, each chromosome consists of two strands called chromatids. They're joined together by a centromere. The two chromatids within a chromosome contain identical DNA-based sequences because each is produced by the semi-conservative replication of DNA. Each chromatid, therefore, contains a complete double helix DNA molecule that is a copy of the single DNA molecule existing prior to replication. Each chromatid will become a separate chromosome once mitotic cell division has been completed. And so, prophase, let's get to figure here. The chromosomes become visible. The nuclear membrane and nucleolus disappear, and the centrioles move apart, forming the spindle fibers. So let's go ahead and take a look here. So we've ended G1. We have paired our sister chromatids together. They're connected with the little central part called the centromere. And now in prophase, they are visible as the chromosomes you're used to. They have the X shapes to them. And then, of course, the sex chromosomes will be X or Y. But mitosis, we've got to divide these chromosomes. So we've got to get rid of this nucleus. And the nuclear envelope has to degrade. So this is a very, vulnerable time for the cell. This is why mitosis also occurs over just a few hours. You can't stay like this forever. And now we are in metaphase, which is the next stage. And so in metaphase, the chromosomes will line up in the center of the cell, and they will attach to the spindle fibers. So the whole time that the nuclear envelope is disappearing, remember the centrioles, which just humans have, just animals have. There's a lot of other animals than humans. But animals have centrioles, and the mitotic spindle fibers will grow from there. And you guessed it, they're made of microtubules. So, with that said, the microtubule is a highway, a railway. Remember learning about that? So now, in metaphase, the whole point is to line up those chromosomes, and we are now going to go into the next stage, where we're going to pull those chromosomes apart. And as you could see, the nuclear membrane at this point has completely disappeared. Anaphase is so neat. The chromosomes are going to be pulled to the opposite side. So the centromeres will split as the spindle fibers shorten and pull chromatids to the opposite side. So the microtubules are going to break apart and pull those chromatids to the opposite side. And those kinetochores are going to be the part that the microtubule attaches to on the centromere. And if you remember, kinetic means to move. We have the kinesins, which are going to be proteins that will move. And kinesins may be involved in this. I'm not exactly too sure. They might be walking these chromosomes apart from each other. I seem to think I've seen that. But regardless, that's what's happening in anaphase. The microtubules are degrading, and the kinesins may be walking those chromosomes away from each other, but we have snapped them apart broken those centromere and pulled them away. And now we have to go through telophase, which is basically the opposite of prophase, in that we are going to rebuild our nuclear envelope. We can see that the chromosomes are going to lengthen now to their relaxed state. They're going to become the chromatin, so that in that new cell they can undergo all those incredible processes of gene expression. And then, if the cell is going to truly divide into two daughter cells, we have to have this cleavage furrow form, which is going to then start to pinch the cytoplasmic contents apart from one another. We've divided our cytoplasmic contents. We've each got mitochondria. We're going to, have a new endomembrane system with the rough and smooth ER, the Golgi apparatus. We'll have our vesicles. We'll have new centrioles, all kinds of cool stuff. And then we'll have the two daughter cells. If the cells don't divide by cytokinesis and we just divide our nucleus, we end up with a larger and larger cell because we've got now multinuclei and we've got larger cytoplasm, and that's called a syncytium. There are some cells in your body that are going to be syncytiums that are formed in this way, such as important cells in your bones. Okay, the role of the centrosome. The structure located near the nucleus of a non-dividing cell. At the center are two centrioles. They replicate in interphase and move away from each other in prophase. Spindle fibers form from the pericentriolar materials around those centrioles and attach to the centromere of the replicated sister chromatids. Other tubules cause the formation of cleavage furrow for cytokinesis. In non-dividing cells, the centrosomes migrates to the plasma membrane and forms a non-modal primary cilium. In ciliated cells, hundreds of centrosomes form and become the basal bodies of the cilia. Ta-da! Interesting. So I wonder, with like other creatures like Paramecium, they have a lot of cilia. And I wonder if they have centrioles. They're what we call protozoans. So they're the ancestors of the animals. All these cool questions. Oh, life. This is why this is my career, my living. Never get tired of it. And I never stop learning. I hope you are the same. That you never tire of this. And you never tire of learning. No matter what you're doing. Particularly if you stay in biology. All right, telomeres. I've heard of telomeres for a while. We take supplements sometimes to help stop the telomeres from degrading because telomeres serve as caps on the end of the DNA, and we see that cells can only divide for so long. And if these telomeres degrade, then we age. So loss of the ability to divide may be due to loss of regions at the end of the DNA called telomeres. Each time DNA is replicated, a little more of the telomere is lost. You die a little more. Damaged telomeres activate p53, which induces cell cycle arrest, senescence, and apoptosis. Cells that can divide indefinitely, such as those in bone marrow, have an enzyme called telomerase that replicates the telomere. So if we do telomere therapy and keep the telomeres long, then hopefully we can, stop aging. However, most cancer cells also produce telomerase. So this is that double-edged sword. So that excess telomerase may be responsible for the ability of cancer cells to divide indefinitely. Telomerase consists of an RNA portion containing nucleotide bases complementary to the telomere DNA, and a protein portion that acts as a reverse transcriptase enzyme, producing telomere DNA using the RNA as a template. Because of the significance of telomeres and telomerase in physiology, cancer, and senescence, the 2009 Nobel Prize in Physiology or Medicine was awarded to 3 scientists who were instrumental in their discovery. All right, so we have hypertrophy now and hyperplasia. Then we'll go into meiosis and we'll be done. Hyperplasia is growth due to an increase in the number of cells, and it is responsible for the growth of most body regions. Hypertrophy is the growth due to an increase in cell size, and it's responsible for an increase in skeletal muscle size. Well, that's cool. Before, we've never really distinguished between the two. We just always called it hypertrophy. But hyperplasia, that makes sense. That's the increase in number of cells. And I guess if it's just hyperplasia, that's normal. If it's dysplasia, that's abnormal. We don't want that, like cervical dysplasia or anything like that. And then hypertrophy is an enlargement of the cells, and that's how muscles get bigger. They don't increase in the number of cell fibers in the muscle. When you get large muscles, they increase in their size, their bulk, their number. All right, I'm going to take a sip of water. Knew this was going to be a long lecture, so I waited for a weekend so I could spend time with it. And again, apologize for the cool little rant there. Don't know if I'll splice that out or not. I'll probably leave it because I want you to be awed by life. Life is amazing, and you're lucky to be here. I'm lucky to be here, and so let's bless ourselves, love ourselves, feed ourselves well, rest ourselves when we can, and when it's time to make babies, if we do, then meiosis is going to be very important. So let's finish with this. So the whole way that life can propagate itself is through meiosis. And for humans, we have 23 pairs of chromosomes, one set from each parent. 22 are called autosomes, and 1 are called sex chromosomes. And the autosomes are for the body. The sex chromosomes, of course, determine if you are biological male or female. So these 23 pairs of chromosomes are called homologous chromosomes. They have the same genes on them, but not the identical DNA. Meiosis is the process by which 2 cell division steps produce gametes, the ova and the sperm, and it only occurs in the gonads, the ovaries and the testes, and it reduces the chromosome number by half. Figure 3.29 shows a karyotype in which chromosomes are arranged in homologous pairs. A false color light micrograph of chromosomes from a male is arranged in numbered homologous pairs from the largest to the smallest. So we see the XY, that 23rd pair there. So that's biological male. So meiosis, as we have learned, has 2 phases. It says that on this slide here, it's a 2-step, 2-cell division step process. So the first one is called meiosis 1, and that's a reduction division. And here we see that in the first division of meiosis, the homologous chromosomes line up side by side rather than single file along the equatorial plate of the cell. The spindle fibers then pull one member of a homologous pair to one pole of the cell and the other member to the other pole. Each of the 2 daughter cells thus acquires only 1 chromosome from each of the 23 homologous pairs contained in the parent. The daughter cells, in other words, contain 23 rather than 46 chromosomes. For this reason, meiosis, from the Greek meion, or less, is known as reduction division. At the end of the cell division, each daughter cell contains 23 chromosomes, but each of these consists of 2 chromatids. Since the 2 chromatids per chromosome are identical, this does not make 46 chromosomes. There are still only 23 different chromosomes per cell at this point. The chromosomes are separated by a second meiotic division. All right, so let's talk about meiosis 1, though, the first one, the reduction division. So the stages of meiosis are subdivided according to whether they occur in the first or the second meiotic cell division. So these are designated as prophase 1, metaphase 1, anaphase 1, telophase 1, and then prophase 2, etc. Or, my dog. They're still in meiosis 1, the reduction division. So prophase 1, homologous chromosomes pair up. Parts are often swapped in a process called crossing over, giving us genetic variation. I'm not going to go on a tangent, but life and diversity on this planet over billions of years. There we go. Boom. All right. Metaphase 1 then is when homologous chromosomes line up in the center of the cell. They line up at random. hence genetic variation. I'm not going to go off on a tangent, but the maternal and paternal chromosomes are shuffled. Genetic variation. Woo, woo, woo. So here we see it. Woo, I get excited. And this is part of figure, not really 3.30. It is 3.31, crossing over. Genetic variation results from the crossing over of tetrads, which are the two pairs of homologous chromosomes, and it occurs during the first meiotic prophase. And then we see a diagram depicting the recombination of chromosomes that occurs as a result of crossing over. So, random assortment of homologous chromosomes and shuffles the maternal and paternal DNA, and then crossing over further shuffles it. And so that's another way that we get all kinds of diversity of life on this planet and within your brothers and sisters and all that good stuff. So then we have anaphase 1. The homologous chromosomes are pulled apart. Telophase 1, homologous chromosomes are separated, and this results in 2 daughter cells with 23 chromosomes each. So this is reduction division. Remember, since each cell now has half as many chromosomes, it's necessary for sexual reproduction, which that's the whole point of sexual reproduction is genetic variation. Otherwise, we could asexually produce, but then we'd be clones, and then we would be susceptible to all kinds of diseases and environmental conditions, etc. So crossing over and shuffling of chromosomes in metaphase 1 results in genetic recombination and genetic diversity. And so then we have meiosis 2. Meiosis 2 proceeds like mitosis with phases prophase 2 through telophase 2. Sister chromatids line up in the center of the cell, and centromeres are broken and pulled to opposite sides. So we're still with 23 chromosomes, but since we've broken up the pairs, now we have 4 cells with 23 chromosomes each. So meiosis reduces the number of chromosomes by half, and then it basically ends in 4 daughter cells. And what is also cool about this is that, particularly in women of multiple species, plants included, 3 of these daughters don't survive. Only 1 matures to become the ovum. All right, so here we see in figure 3.30, meiosis or reduction division. The first cell at prophase shows that the DNA has been duplicated to form homologous chromosomes. 2 homologous pairs are shown, with the chromosomes of each pair shown red and purple. In the first meiotic division, the homologous chromosomes of a diploid parent, which means they have the full set of chromosomes, that pair from mommy and that pair from daddy. That's what diploid means. So that's the 46 total, 23 pairs diploid. So anyway, the homologous chromosomes of a diploid parent in the first meiotic division, they are separated into the 2 haploid daughter cells. Each of these chromosomes contain duplicate strands or chromatids. And so that's where we end right here, telophase 1. Okay, so these are daughters, and they are haploid because they don't have pairs of homologous chromosomes anymore. They just have like 1 homologous chromosome, okay? And now what we're doing is we're going to split those bad boys. We're going to split the homologous chromosomes apart into their sister chromatids. And then we are going to basically end up with 4 daughter cells that each have like a haploid chromatid. So we went from diploid chromosomes, you know, just a full pair of mommy and daddy, all right? And then we split those into haploid chromosomes. And then remember, once the DNA, you know, is in the chromosomes, it's 2 sister chromatids. Now we got to get those sister chromatids apart. And that's what the whole point of meiosis 2 is. And so we have individual chromatids here in our daughter cells. And, you know, like I said, with particularly in a female, 3 of those aren't going to survive to become, you know, eggs. Only 1 will. So we are all survivors. Okay, so this is table 3.3 that is summarizing the stages of meiosis. And hallelujah, we're like at the end of the chapter. This is like my favorite stuff. And there's a whole class on this, cell and molecular biology. And again, it's what made me become a biologist. Some of y'all be like, this is what scared me away from biology. But when you really think about how incredible these processes are. It's just unbelievable, and it just makes life just even so much more beautiful and sacred. So here we see in table 3.3 those stages of meiosis. Prophase 1, the chromosomes appear double-stranded. Each strand, called a chromatid, contains duplicate DNA joined together by a structure known as a centromere. Homologous chromosomes pair up side by side. So in this case, we're not going to make, daughter cells that are identical in numbers. We're going to do reduction division. So once we pair them up side by side, now we've got to split them apart. So in anaphase 1, the homologous chromosomes separate. The two members of a homologous pair move to opposite poles. In telophase 1, cytoplasm divides to produce two haploid cells. In the second meiotic division, in prophase 2, chromosomes appear, each containing two chromatids. Now we got to get those chromatids apart. So metaphase 2, the chromosomes line up single file. So this is a lot more like mitosis. And we're going to then split the chromatids apart. But there's only 23 that were split, not the whole 46. And so we're still haploid. And in telophase 2, the cytoplasm divides to produce two haploid cells from each of the haploid cells formed at telophase 1. All right. So if you thought that was the end of the story, it pretty much is. But there's the epigenetic stuff, which is a whole new branch of science. You can be an epigeneticist. And so silenced genes are passed to daughter cells without a change in DNA-based sequences, but there can still be changes in gene expression. So mechanisms of epigenetic inheritance occur outside of the genetic sequence itself. That's what epigenetic means. So we see that there's post-translational modifications of histone proteins. And then here's one that I'm super familiar with. There's methylation of cytosine bases in the DNA that precede guanine, and that decreases gene activity. So it says, because such heritable changes in gene expression do not involve changes in the DNA-based sequence, they constitute epigenetic inheritance. And so, yes, they are inheritable. And says that genetic inheritance is determined by the sequence of DNA-based pairs in the chromosome. However, not all of these genes are active in each cell of the body. Some genes are switched from active to inactive and back again, as required by a particular cell. Activity of these genes is subject to physiological regulation. Other genes may be silenced in all the cells in a tissue, or even in all the cells of the body. Such long-term gene silencing occurs either in the gametes, and so is inherited, or in early embryonic development. The silencing of these genes is carried forward through cell division to the daughter cells. Other mechanisms involving post-translational modifications of histone proteins increase gene expression and are carried forward to daughter cells. Because such heritable changes in gene expression do not involve changes in the DNA-based sequence, they constitute epigenetic inheritance. And then the epigenetic inheritance occurs by two primary mechanisms. We just saw that one is post-translational modifications by acetylation, and the other is the methylation of cytosine bases. All right, so the last paragraph says that scientists were surprised to discover that some epigenetic changes can be transmitted to the next generation through modifications of the germ cells, which are the sperm and the ovum. During germ cell development, there are mechanisms to eliminate epigenetic changes in the stem cells of sperm and ovum so that the chromosomes of the fertilized ovum start fresh. However, some epigenetic changes caused by food deprivation, for example, have been reported to be inherited by the offspring. The biological nature of epigenetics and its possible medical applications, for example, in cancer detection and treatment, are active areas of research. I'm shutting my book, and I'm stopping the recording. If you made it through this with me, you are a trooper, and I am truly impressed. And blessings to you all. Have a great day or evening, wherever you are.